देख लेना ट्रेनिंग की मेरी सारी प्रैक्टिकल लाइव कर दो. Welcome back, Dr. Gaur, and now we'll request you to please start from the second half of your training session, please.
Thank you, ma'am. We will discuss about the blood composition, function of blood, and how to test any blood specimen. So these are learning outcome of this chapter. Definition of blood, differentiate blood component, classification of blood cells, and also classification of plasma proteins and functions of bloods. It is a blood. What is blood? It is a fluid connective tissue. Why it is connective tissue? Because it connect various organs of human body, and it transport various nutrition product also. It transport also excreted product also. So it is called connective tissue. It is like in a fluid situation, but it's called a connective tissue. It makes up to 7 to 8% of total human body weight, and for the healthy 7 kg person, its volume should be 7 liter. Sorry, 5 liter. And it is made up of two components: cellular material and liquid components. Liquid component is a plasma, and cellular component is a type of blood cells. There are blood is a circulating tissue consisting of three type of cells, which is called first one are blood cells, which is also another name is synonym, and erythrocyte, and second one is white blood cells, that is also called as leukocyte also, and third one is the platelets, and which is another name is thrombocytes. The cell listed above are suspended in a liquid known as plasma. So there are two components for the cellular part, and second one is the liquid component. How form the blood in human system? That is called hematopoiesis. The formation and development of blood cells occur in the bone marrow, and in adult, the cellular elements are produced in the bone marrow. This is a continuous process. After aging of blood cells, these cells destroy in the endothelial system, and new cells transport by the blood and from the bone marrow and from the human system. Some WBCs are produced in the lymphatic tissue and bone marrow also. So blood cells also form in the bone marrow and lymphatic tissue. Blood cells need certain nutrient to form properly. So these include iron, folic acid, vitamin B12. So these type of nutrient this is required for the formation of blood cells. So every needy person, doctor suggests iron tablets, folic acid, and vitamin B12 in the requirement of access of blood. Uh, all blood cells form some. All blood cells form come from hematopoietic stem cell. These are stem cell which is present in the bone marrow, and all the human cell are originate from the stem cells. Stem cells having its ability to become any type of cell. These cells can become any blood cells. This is the composition of whole blood. First, I already explained the cellular part and liquid part. So this is the type of some whole blood, and it consists of 40 to 46 to 63% of plasma, and remaining part, which is covered form element, which is 37 to 54%. It may be up and down according to condition. Maybe cellular component may be more or less. Then correspondingly, plasma concentration will be increased or decreased. Out of 46, 60% of plasma protein consists 7 part and other solute are 1%, but and out of 60%, it consists 98 to 92% of water, which is transport organic and inorganic molecules, form elements and heat also. So we can discuss in two component broadly, plasma protein and other solute part. Plasma protein, out of whole 7% of plasma protein, 60% part consists albumin. This is a major contributor to osmotic pressure of plasma, and it transport lipids, steroids, and other hormones also. Second plasma protein is a globulin, which form in globular form, which transport ions, hormones, lipids, and immune functions also. Remaining part is the fibrosis is a type of also plasma protein. It is essential component of clotting system, which can be converted to insoluble fibrin in when when bleeding occur. And other regulatory proteins which is less than 1%. In this list it consists of various type of enzymatic protein, proenzymes, and hormones are considered in this plasma proteins are major part of protein albumin and globulin, and remaining part of fibrinogen and other regulatory proteins. And other solute of other in component of other solute 1%. First one electrolytes, normal extracellular fluid, ions composition essential for vital cellular activities. Ions contribute to osmotic pressure for body fluid. Major plasma electrolytes are there: sodium, potassium, calcium, magnesium, and electrolytes in negative form, chloride, bicarbonate, phosphate, sulfate ions are there. So these are electrolytes which manage the electrolyte balance of human body and acid base balance of human body blood system. One human blood system acid base balance is imbalanced, then these electrolytes are manage everything, either pH increase or decrease, either osmotic pressure increase or decrease. And second factor is organic nutrient. It may be used for ATP production, other growth and maintenance of cell. This kind of other nutrients are also available in the solute form in plasma. Organic waste also present in the 1% of solute, which is on the soluble form, just carried to site of breakdown or excretion, include urea, uric acid, creatinine, bilirubin. Ammonia ions also solute in the solute form of plasma protein, which is used for the transport. And this part showing all the plasma composition of blood, and this is this slide for the four elements of blood component. So form element, which is 40% around, and it consists three main component. First, platelets, WBC, and red blood cells, in which out of 40%, all 99% consist of blood cells, which is similar to like this one. And second are platelets, which is also name of thrombocytes, and white blood cells. White blood cells, five type of cells are there. First one, neutrophils, eosinophil, basophil, lymphocytes, and monocytes. In number of cells, yeah, the first more number of cells are present in form of neutrophil. Second, lymphocytes are present around 20 to 30% are there. Eosinophil 2 to 4% are there. Monocyte 2 to 8% are there. And but basophils are less than 1% in human blood system. These are specific functions which can be discussed in later. So this is made of cells that are suspended liquid, which is called plasma. And plasma make up to 55% of human blood. Plasma is made of 90% of water and 10% protein part, which we have already discussed, in which lipids, carbohydrate, amino acid, antibodies, hormones, electrolyte, waste salt, and ions are. Everything are in form of nutrient or either in form of accelerated product, which is also transported via the which is also form of accelerated product, which is all accelerated product and in a nutrient product are present in the plasma, and plasma can be work as a transport media for the both of them, either nutrient or media or either accelerated product. Blood cells make up of the remaining 45% of blood. RBC make up of 95% of the blood cells. White blood cells and platelet make up of other 1%. Each type of blood cell perform a different function. RBCs are red blood cells looking like this one, and this is the imaginary picture, pictorial picture for the how RBC appear, how RBC views. WBCs are looking like this one, and platelets thrombocytes are looking like this one. One by one, we will discuss RBC, red blood cells. Another name is erythrocyte. Most evident cell in the human blood and mostly present in human blood in form of RBC, number of 4 million to 6 million per microliter of blood. 1 microliter of blood they found in 4 million to 6 million quantity. This is the main cell which is formed in the bone marrow and transported from the bone marrow to the blood circulators. And main function of RBC is the transporting oxygen and carbon dioxide both way. It mature form don't have a nucleus. This RBC don't have any nucleus. This you can see in the picture. The central part is black without nucleus. Earlier phase of RBC, these have uh, nucleus, but later on they mature in form of RBC. The RBC and the nucleus has been disappear. So this shape as a biconcave disc may be found in biconcave disc, and this is a 6 to 8 micrometer in diameter. This stain pink tan and center of cell is lighter center area of pallor. So mostly peripheral area consists having more hemoglobin concentration. Hemoglobin is found in the RBC, which is iron protein, which is mostly important for the human health. And it carries oxygen from the lungs to the rest of the human body and also collect the carbon dioxide from the outside the human, all the body and transport to the lungs. And carbon dioxide bind to the RBC and taken to the lungs to be exhaled. So it is the most transport carrier for both oxygen as well as carbon dioxide. How it oxygen carry over oxygen bind with the hemoglobin and transport in form of oxyhemoglobin, and when carbon dioxide transport this bind with the hemoglobin and make carbo hemoglobin. Second, most cells of blood, which is white blood cells, this is the leukocyte or WBC. This is the largest size of blood cells, lowest number of blood cells, just like in 1 microliter, it may be found in 4,000 to 11,000 per microliter. These cells also form in bone marrow, but and some cells of WBC form in lymph glands also. Primary cell, this is the primary, WBC is the primary immune primary cell of immune system. This is the first line of defense. The WBC is called as the first line of defense. Fight disease and foreign invader. Any factor which is entering in the human body, which is the foreign invader, it can be neutralized by the WBC. It contains nuclei with DNA. So these cells having a perfect size of nucleus and particular DNA material also, and the shape depend on the type of cell. These certain WBC produce antibody. So in which one or two cells of WBC produce WBC antibodies also. Lifespan is from 24 hours to several years of this, and RBC lifespan is 120 days also. Size is WBC size is 8 to 20 micrometer and RBC size is 7 to 8 micrometer. So WBCs are greater than RBC in size. There are two different type of WBCs. First from granulocyte and second one is the agranulocyte. Granulocyte cells having some granules in the cytoplasm of cells, and but agranulocyte does not having any granules in the cytoplasm. So in granulocyte, three type of cells are there: neutrophils, eosinophils, and basophils. Neutrophils, eosinophils, and basophils are you can see in the picture. First one is the neutrophils. It can be appearing multi-lobe nucleus, and eosinophil is the like two lobes that there, and basophil is the multi-lobes there, but nucleus shape in kidney structure. Agranulocyte, you can see the lymphocyte and monocytes also there. These cells do not have any granule in cytoplasm. Another third type of cell is a platelet. These called thrombocyte or platelets also mostly important in the clotting system. These cells also form in the bone marrow and they can be fragmented from the cytoplasm of megakaryocytes. These are smallest of blood. These are smallest of blood cells, and size of platelet may be 1 to 4 micrometer in diameter. This shape can be round, oval, or appear spiky. Lifespan of the platelet lifespan is 8 to 12 days. So after 12 days, they may be disappear, destruct, and new platelets continuous form from the bone marrow. These involved in the clotting process, and platelets seal, bound, and prevent blood loss also. This is a main factor which is involved in the clotting process. These help repair damaged vessels. Normal range of platelets should be in human blood 1.5 to 4.5 per microliter of blood. These platelet can be stained in bluish with reddish or purple granules found in the peripheral blood. And functions of blood. First one is the respiratory, which is the most important done by the hemoglobin, which is found in the RBC. Transport of oxygen from lungs to tissue and carbon dioxide from tissue to lungs. Second most important function is the nutrition. So these convey absorbed fluid material from alimentary canal to the tissue for utilization and storage purpose, and also supply all food material from the all organs of the human system. Third one, excretory. These transport of metabolic waste to kidney, skin, and intestine for the removal of excreted product. And homeostasis, what is the homeostasis? Is a control mechanism of homeostasis. Homeostasis for water, pH, electrolyte concentration. These blood can be managed everything in case of water deficiency or water increase, pH increase or pH decrease, electrolyte concentration increase or decrease. So homeostasis can be managed in both conditions. And blood can manage body temperature also. These are the functions of human blood. And what kind of function of plasma protein? These protein are useful in blood coagulation. Albumin is useful in the maintaining. Fibrinogen is useful to maintain the viscosity of blood, which is most plasma proteins, and most clotting factor, which is useful to maintain viscosity of blood, and it may be useful to maintain the acid-base balance in the body, and plasma protein act as a buffer. I already told you the buffer. Blood can work as a like buffer, and it can be managed in acid-base balance, either acidic pH of blood or either basic pH of blood. So this can be blood can be maintained. Plasma proteins can be maintained acid-base imbalance. Immune function, gamma globulin produce antibodies which provide immunity. So these plasma proteins also uh, help in the provide immunity. The first part of the training has been completed, in which we have discussed earlier about the normal definition of blood, types of blood, and how cells form in from the bone marrow, and the plasma composition of blood, which consists of plasma protein and other solutes are there. And second, form elements in which the three components are there: platelets, WBC, and white blood cells and red blood cells are there. In which RBC, red blood cells are abundant in number, 98%, white blood cells are less than 0.5%. So these are cells. There are three type of cells are there, and RBCs, concentration of plasma is there, detailed description of RBCs, number, shape of RBC, lifespan of RBC, and what is the function of main RBC. RBC having an hemoglobin concentration works is there, and WBC is what kind of number of WBCs should be in human being, and what is the main function of WBC, and the classification also WBC into form of like granulocyte and agranulocyte. And granulocyte, three type of cells are there according to nucleus structure: multi neutrophil, eosinophil, basophil, and agranulocyte is having two sides, lymphocyte and monocytes also. And third type of cells, platelets are there. Platelet number of cells, 1 to 5, number of cells lifespan 8 to 12 days, 1 to 5, 12 to 4 micrometer in diameter, and 1.5 to 4.5 micrometer. That's the normal range of blood, and function is the main clotting purpose. And these are conclusive function of all human blood, which is 5, and sixth one is the plasma protein function. That is okay. Next step, next part, we'll discuss about how the test, how the estimate the hemoglobin, how the estimate of leukocyte, total WBC, and how the estimate of platelets also. Hello, hello, hello.
Yes, sir.
Ma'am, first part completed.
Sir, please continue. We'll break off for lunch once it gets over now.
Okay.
The people have joined on YouTube, so I would request the participants to start placing your queries. Once the training gets over, then I will put up your questions to the expert.
This is the chapter of estimation of hemoglobin, only hemoglobin estimation. So these are objectives after this chapter. So how can we describe the structure of hemoglobin, the list of function of hemoglobin, and explain the various laboratory methods for estimation of hemoglobin, and enumerate the advantage and disadvantage of each method. And we can discuss the normal value and interpretation of a normal result also. So hemoglobin is a tetramer consisting of two pairs of similar polypeptide chain called is globin chain, and each of the four chain is attached heme, which is complex of iron and ferrous form or protoform. So out of the whole concluding is the hemoglobin is consisting two pair of globin chain and one group of heme. Heme plus globin, this is called hemoglobin. Based on 96% type of hemoglobin present in adult is called HbA, and it has two alpha globin chain and two beta globin chain. This is the another structure description of hemoglobin. We are not discussing about the detail again, alpha 2, alpha 2. So this is a summary of chart, the type of hemoglobin. So first adult HbA, which will be found in the adult only, structure two alpha chain of globin and two beta chain of globin, which is found in adults. And another type of protein HbF, which is called fetal as hemoglobin fetal HB, and it consists two alpha chain and two gamma globin chain, and present in fetal life also. HB Portland, Delta, Gamma, and embryonic life, and Gover 1 and Gover 2, Epsilon, Alpha 2. This is another abnormal hemoglobin also, but we found only 99% in HbA, which is at adult hemoglobin. Function of hemoglobin already described in previous part of training. Heme has the ability to bind oxygen reversibly and carry it to the tissue. It also facilitates the exchange of carbon dioxide between the lungs and tissue. The hemoglobin function is the primary medium of exchange of oxygen and carbon dioxide. Without presence of oxygen, hemoglobin, we can't exchange of gases. So there are various methods of estimation of hemoglobin. Sahali's acid-hematine method, cyanomath hemoglobin method, oxyhemoglobin method, alkaline hematine method. So there are various kind of method. We will discuss two most important, Sahali method or cyanomath hemoglobin method. Commonly used method are Sahali acid-hematine method, and this is called also cyanomath hemoglobin method. Details of this method are described. So Sahali method में what is happening? So also we discuss in the section. Principle, what is the principle of the Sahali method acid-hematine? It is mixed, blood is mixed with the N by 10 HCl, resulting in the conversion of Hb to acid-hematine, which is brown in color. The solution in diluted till its color matches with the brown colored gases glass of the comparator box. The concentration of Hb is read directly. What kind of equipment required for this purpose? This test hemocytometer. Also purpose required. Hemocytometer consists of comparator box which has brown color glass or either side. HB pipette which is marked up to 20 mm, 0.02 ml blood is required. Tube with marking of HB on the one side. Glass rod is required and dropper is required. And reagent and by 10 SL required, distributor required. And sample, venous blood sample collected in EDTA, that's the EDTA, ethylene diamine tetraacetic acid, which is anticoagulant, which prevent the blood from the clotting. What is the procedure for estimation of HB? Add N by 10 HCl into the tube up to mark 2 with the additive sample by Janssen inversion. Fill the pipette with 0.02 ml blood and wipe out the external surface of pipette to remove any excess blood. Add the blood into the tube containing HCl. Wash out the content of the pipette by drowning in or blowing out the acid 2 or 3 times. Mix the blood with the acid slowly. Allow to stand undisturbed for 10 minutes and place the hemoglobinometer tube in the comparator and add distilled water to the solution drop by drop, stirring with the glass rod till its rich color matches with that of the comparator glass. While matching the color the glass rod must be removed from the solution and held vertically in the tube and remove the stirrer and take the reading directly by noting the height of the diluted acid material and express in gram percent. So this one you can see the both side the comparator box color box is there, and this is the box, and you place the your hemoglobinometer inside in between. So you can remove all the stirrer and glass rod from the hemoglobinometer, and you can read the reading after standing position, which should be noted in the hemoglobin in gram in percentage. What kind of advantage of this test? Is it this test is easy to perform, quick and inexpensive, and can be used as a bedside procedure, which is called POCT, point of care testing. We can perform anywhere. We need sample, and this test is required. Does not require any technical expertise. We can explain personally, and we can conduct. What kind of disadvantage of this test? This is less accurate. All hemoglobin, just like type of hemoglobin, oxyhemoglobin, sulfur are not converted into acyl. Hence, the value of hemoglobin obtained is less than the actual value. The color of acyl develops slowly, so it takes too much time. Color of acyl faded with the time, and dilution must be done exactly after 10 minutes when the color development is maximum. So it should be completed within 10 minutes. Individual variation is matching of color is seen. So various color of other disadvantage is there. If the matching point is passed, the whole procedure has to be repeated. If matching point where all both comparator color and hemoglobin color matches, if that color is point is passed, then you need to be repeat again. Color of glass in the comparator box tend to fade with the time. So whatever, if we are using old hemoglobinometer, that is difficult to read the reading in proper way, and lack of true strand standards cannot be applied, so we take the reading without the use of standard. Second method is the cyanomet hemoglobin method. This is the internationally recommended method for the determining hemoglobin. What is the principle of this test? It is blood is diluted in the sodium solution containing potassium cyanide and potassium ferrocyanide. The latter convert Hb to meth hemoglobin, which is converted to cyanomet hemoglobin by potassium cyanide. The absorbance of the solution is then measured in a spectrophotometer at a wavelength of 540 nanometer in a colorimeter using a yellow-green filter. This is the principle. What is the principle? We mix Drapkin solution. It is called Drapkin solution, and Drapkin solution of blood is mixed, and the color pigment is formed, which can be read out in the with the help of colorimeter. What kind of equipment is required? HB pipette is required, spectrophotometer or other colorimeter is required, reagents required. What is reagent? Drapkin solution is required, which has pH 7 to 7.4. And what kind of ingredient of Drapkin? Potassium cyanide 50 mg is there, potassium ferrocyanide 200 mg is there, potassium dihydrogen phosphate for 140 mg is there, and non-ionic detergent 1 ml and distilled water 1. This should be mixing within 1 liter. This is ready to use available in the market in name of Drapkin solution. But it consists of potassium cyanide, which is highly potassium cyanide is the you know about the nature of potassium cyanide. This is poison, so it should not available in the market without prescription or without authenticity. The solution should be clear and pale yellow in color when measured against water as a blank in spectrometer at wavelength of 540 nanometer. So first we need to check the appearance of solution, and uh, absorbance of solution should be 0, that is blank, at 540 nanometer in the spectrophotometer either colorimeter. The absorbance must be 0. The solution is unstable if exposed to light, can be stored at room temperature in brown borosilicate bottle for several months. However, if the room temperature is higher than 30 degrees centigrade, the solution should be stored in the refrigerator but brought to room temperature before use. So before test, need to maintain the temperature of reagent, and it should never be frozen. pH of solution must be checked every month. Discard the solution if found to be turbid or pH is outside range, its absorbance is not zero. So we can't use this solution for the test. And don't prepare Rapkin solution by mouth because it is very high, highest level of poison. And what kind of sample is required for the SB estimation? Venous blood collected in the DTA. What is the procedure of this principle? What is the procedure of this test? First, take the 5 ml of Drapkin solution in a test tube, label it, and mix the blood sample by junction inversion and drop 0.02 ml of blood into the HB pipette and wipe the outer surface of pipette to remove excess of blood. Place the pipette into the tube containing Drapkin solution and slowly expel the blood into the solution. Mix well and let it stand undisturbed for 5 minutes. Wait for 5 minutes and measure the absorbance of this solution at 540 nanometer in spectrophotometer after adjusting the optical OD at zero by using Drapkin solution as a blank. We need to we need to read blank solution zero, then we can measure the test absorbance. Calculate the hemoglobin concentration using standard curve. What kind of advantage and disadvantage of this test? All form of HB except sulfur hemoglobin are converted. So all form of hemoglobin can be converted in this test. So this test is mostly suitable. And visual error is not there. So no color matching is required. Cyanomath hemoglobin solution is stable and its color does not fade with the time. So reading may not be taken immediately. We can take reading by some time later. And absorbance may be measured soon after dilution. A reliable, stable reference standard is available from WHO for direct comparison. We can test first we can test control first, and then we can test. We can take measurement of test sample. What kind of disadvantage of this test? Diluted blood has to stand for a period of time to ensure complete conversion of HB. So this test take more time. Potassium cyanide is a poisonous substance. That is why Drapkin solution must never be pipetted by mouth and not available in the market by the unauthorized person. It is available for the authorized person. The rate of conversion of blood containing carboxyhemoglobin is slow considerably. Prolonging the reaction time to 30 minutes can overcome this problem. Abnormal plasma protein cause turbidity when blood is diluted with the Drapkin solution. So a high leukocyte count also causes turbidity on dilution of blood. Centrifuging the diluting blood can help overcome the turbidity. We can centrifuge before the test the sample if samples having any turbidity. How can prepare a standard curve for hemoglobin estimation by cyanometer hemoglobin method? In laboratory, we test several samples in day. It is more convenient to prepare a standard curve for HB. How can we prepare the standard curve? At WHO International Reference standard is available commercially as 10 ml seal sample. This solution is stable for years. The exact concentration of HB present in the solution is indicated as label. We can plot the graph, one x side concentration of HB and y axis on optical density of the solution, which will be read out the device. These are two most important method. Oxyhemoglobin method is also available in the market. HB is converted to oxyhemoglobin by reaction with ammonia, and the color of the solution is measured in a photocalorimeter. So these methods are not up to the mark and not related to the field. And these only two methods are important, and two methods are working in the laboratory for the diagnostic purpose also there. So second part of this training session has been completed. In this training, estimation of hemoglobin, the type of hemoglobin, and the type of methods of estimation of hemoglobin. Two methods are most important, which can be estimate hemoglobin: Sahali method and cyanometer hemoglobin method. So this is the second part completed. We can go on third part.
Dr. God, there are some queries on the YouTube. Just let me check.
जी मैम.
Uh, they want a small recap of the estimation method that you have just spoken about. So just a small recap.
Small cap of WB hemoglobin estimation.
Yes.
Okay.
Please go ahead, sir. A small recap only.
Okay. So two methods are important here. First one, Sahali method. Sahali method. Principle of Sali method in this way, we mix N by 10 HCl with the blood, and a second product is formed, which is called acid hematine, which is in brown in color. The solution is diluted till its color matches with the brown color. So what we do in the this test, we mix blood sample and N by 10 HCl in a container, given container, and later on we added diluted some distilled water up to the mark when comparator box and test box, both color are mixed, both the color are reaching in equal. These are equipment. Comparator box we require, HB pipette is required, tube with marking of HB on one side. So this tube required for main purpose. We mix N by 10 HCl and hemoglobin and water. We mix in this tube, or three component will mix in tube. First, we will add N by 10 HCl. Second, we will add hemoglobin, sorry, blood, and third, later on we will mix distilled water drop by drop. So these N by 10 SCL required, distilled water is required, and venous blood collected EDTA is required. First, add N by 10 SCL into tube up to the mark 2 percentage. Mix EDTA sample by Jensen inversion with the help of RBC pipette, and add the blood into the tube containing HCl. Wash out the content. At the blood, wash out the content of the pipette by drawing in the blowing out. So drawing it out or drawing blowing out we can do, and twice or three times. So all as hemoglobin can be, all WBC, or sorry, blood can be pass out from the pipette, and allowed to stand undisturbed for 10 minutes. Then place the hemoglobinometer tube. This is the hemoglobinometer tube. So hemoglobinometer tube place in the comparator box. Comparator, what is the comparator box? You can see the in picture. This is the comparator box, and this is the hemoglobinometer tube. And first we place the tube inside in this place, and we can add drop by drop water. And after adding drop by drop, the color of hemoglobinometer should be matched with the comparator box. So all three colors should be equal. Then stop the adding of blood and remove the stereo or mixer, so we can read the actual reading of the hemoglobin. So don't mix distilled water in frequent way. Add distilled water slightly, one by one drop. One drop adding, then mix, and one drop add, then mix, one drop add, then mix. So we can compare the color of hemoglobinometer and comparator box. Okay, so then remove the stirrer and take the reading directly by noting the height of and hemoglobinometer linings for the reading. This test is easy to perform, quick, inexpensive, can be used as bedside procedure, which is called POCT. Anywhere we can perform. We need only minimum requirement specimen, but it has lot of disadvantage, less accurate. All type of hemoglobin cannot convert it in the acid hematine. So we found the reading in actual way is not correct. We found the reading is less the actual way. The color of acid hematine develop very slow. Color of acid hematine fade with the time. So we take the reading after a lot of time, a couple of time. So reading will be false, negative or positive. individual variation in matching of color is seen if person who is performing the test, if he or she having any color competing disorder, or so actual reading could not be taken. If the matching point is passed, the whole process has to be repeated, and the color of glass and comparator box extend to old comparator box should be avoided for this test. Second method is Sinomath hemoglobin method, which is internationally accepted. What need to be done in this HBP required HBP spectrophotometer reagent Drapkin solution, and what we need to follow: take 5 ml of Drapkin solution in a test tube, normal test tube, which is vacuum use solution in test tube. Mix the blood sample by Janssen version and draw 0.02 ml, 20 microliter of blood into the HB pipette. Wipe out the outer surface of pipette to access remove blood. Place the pipette in the tube containing Drapic solution. Slow mix well, and after mixing of blood and Drapkin solution, you need to read the absorb optical density of mixing sample. So, what is the difference between both tests? In Sahali method, we mix N by 10 HCl blood sample plus distilled water, and in there we mix Drapkin solution 5 ml plus blood sample only. We don't require adding any distilled water, and directly we can calculate. Directly we can read the absorbance through the with the help of spectrophotometer or the colorimeter is whatever is available in the laboratory, and calculate with the standard curve method. And these are advantage and disadvantage there. So various advantage of this cyanomethamoglobin is there.
Sir, I think we can go for 5 minutes break, and then we can come back, and then we'll finish off the training before we go out for lunch. In the
Okay.
meanwhile, I will request participants to place their queries on the YouTube. We will be meeting at around 1:35. It's 1:30 right now. We'll come back at 1:35. Thank you.
Okay. Dilution fluid is increasing, so we can this more